Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Today, me, Aina Rasida, along with my fellow friends, Aina Atikah, Nurhanin and Umu Afifah, are going to present the article assigned to our group entitled Environmental Epigenetics Crossroad between Public Health, Lifestyle and Cancer Prevention. This video will be comprised of 4 main subtopics, which are the mechanisms on epigenetic regulation, the influence of environment on epigenetics marks, briefly on the role of epigenetics in chemo prevention, and lastly, the conclusion and the Islamization part. The first mechanism which regulates the epigenetic is DNA methylation and hydroxymethylation. This process is mediated by DNA methyltransferase, or well known as DNMT. There are several examples which are responsible to maintain the genomic integrity, which are DNMT1, DNMT3A, and DNMT3B. However, this process is actually mainly altered in the cancer cells, which lead to genomic instability. The second mechanism is histone modification. Normally, the modification process will take place at the histone tail. For acetylation process, it is mediated by HAT, which is known as histone acetyltransferase, while the acetylation process is mediated by HDAC, which is known as histone deacetylase. Other examples of histone modification are phosphorylation, ubiquitination, ADP ribosylation, and biotinylation. The third mechanism is chromatin remodeling. Chromatin remodeling may cause cancer as it may activate and suppress the gene transcription. The chromatin remodeling can be achieved by several means such as ATP-dependent protein complex formation, histone modification by polycomb protein (PCG), interaction of non-coding RNA (ncRNA), and histone acetylation or histone deacetylation process. The last mechanism which regulates the epigenetic is non-coding RNA. Non-coding RNA is the RNA which is not translated into protein. Generally, non-coding RNA interfere with other RNA, and this mechanism is called as RNA interference. This mechanism is considered as epigenetic as it does regulate the gene expression without altering the gene sequence. One of the main examples of non-coding RNA is microRNA. In cancer, the microRNA is deregulated and it leads to silencing complex formation.
There are 3 factors that can influence environment on epigenetic marks. The first one is lifestyle and epigenetics. Smoking and high alcohol consumption indeed can modify the methylation pattern of tumor suppressor gene. Cigarette smoke contains a full spectrum of carcinogenic substances. However, there are no remarkable results and consensus regarding the effect of smoke in DNA methylation. During early stages of lung carcinogenesis, we can see that methylation patterns of tumor suppressor genes has been modified. For alcohol consumption, pregnant mother who consume alcohol can disrupt the neuronal growth and development of the baby. There are 84 genes involved in brain metabolism and differentiation. Usually, genes are modified epigenetically by acute alcohol administration in human homolog. These often involve in neurological pathologies. There are high possibility of gene methylation modified. However, in one experimental studies of direct carcinogenic activity of alcohol failed to show any remarkable results. The main reasons are methylation pattern of tumor suppressor gene and DNA repair genes altered in colorectal cancer patients. And the second one is due to high alcohol consumption and low folate intake. Nonetheless, significant association between folate intake, alcohol, and methylation MLH1 failed to be demonstrated.
The second factor is endocrine disrupting chemical.
EDCs, or endocrine disrupting chemicals, are chemicals that mimic the action of natural molecules. The accumulation of these chemicals will alter the endocrine signaling pathways with various subsequent effects on epigenome. Examples of EDCs include BPA. BPA is used in the manufacture of epoxy resin. It is commonly consumed by humans through the consumption of canned foods. This compound can alter the expression of selected genes via histone methylation, microRNA expression, and changes in DNA methylation. Another examples are DDT, insecticides, and pesticides. These EDC compounds can alter DNA methylation, the level of DNA methyltransferase, which is the DNMT3B expression, as well as affecting the CpG methylation levels. Another example of EDC is DES, which is a synthetic estrogen blocker. The in utero exposure to DES was found to alter the expression and methylation of genes which are relevant for certain cancer development.
Influence of nutrition on epigenetics. Certain diet components can modify epigenetic pattern and act on DNA methylation or histone modification. Also acts as exogenous microRNA. For example, insufficient or excess of vitamin B6, B9, and B19 can modulate DNA methylations. Selenium is an essential diet component which can inhibit the NMT and HDAC. Also reactivates the gene expression in response to oxidative stress and protect from carcinogen. Next, polyphenols, which found in green tea and vegetables, can interfere with the NMT, HDAC, and HAT activities, as well as revert malignancy-associated epigenetic alterations in cell line. Lastly, phytoestrogens, mainly in soybeans, can inhibit with the AMT and HDAC, thus reduce risks of hormone-related tumors. Psychoepigenetics. Early life experiences are perceived as the building block for adulthood, so childhood abuse or suicide victims experimentally by exposing the rats to stress may result in alterations of methylation levels of neuronal glucocorticoreceptors.
It is proposed in the article to develop a synthetic drug which serves as a therapeutic drug in cancer. It is mainly targeting the epigenetic pathway by inhibiting the DNA methylase and histone deacetylase. The example of synthetic drug used to inhibit the DNA methylase action are 5-aza-2-deoxycytidine and 5-azacitidine. The synthetic drug used to inhibit the action of histone deacetylase are suberolenolide hydrosamic acid, or known as SAHA, and trichostatin A, which also known as TSA. It is also mentioned in the article that there are several examples of natural bioactive food components which act as chemopreventive agents by involving in the DNA methylation process. Examples of bioactive compounds are polyphenols, retinoid, and phytoestrogen. These compounds can be mainly found in the vegetables and the fruits. It can be concluded that environmental and lifestyle factors do have impact on epigenetic regulatory processes, and as a take-home message, in line with the saying of prevention is better than cure, one should be able to monitor their nutrition intake, lifestyle behavior, as well as their surrounding factors, as those factors may interfere with cancer development via the epigenetic mechanisms.
For Islamization, from Surah Yunus verse 101 and Surah Ar-Rahman verse 7-9, cancer is mainly due to dysregulation of normal cellular function, so it is an obligation for all Muslims to seek knowledge and acquire more understanding on carcinogenesis. As such, reverse the epigenetic change and create epigenetic balance may have a therapeutic potential for cancer.